to Chapter 6, Microbial Growth. A lot of this is going to build upon the knowledge that you've already been learning so far in this class. So that's exciting, and we'll do at least 2 parts, perhaps even 3 parts. We'll see how long it ends up going. So, of course, before we start diving into the content, want to remind you the reason why it's so important to study all the different aspects of microbial growth, right? Of course, part of it's just to study and learn about these organisms and get a better understanding of the life around us. But in this class, our big concern is to understand diseases, so that we can prevent them, so we can prevent the spread throughout the community, so we can make vaccines, and then when people do become ill, we can treat it. We also like to apply these principles to, slowing down food spoilage, preventing damage and spoilage to crops, increase the efficiency of our crops and stability of our food supply. We also like to understand how to produce molecules like enzymes, or even the molecules that are used for insulin, right? And other industry and medicinal manufacturing, right? So by understanding these components of microbial growth, it allows us to do all of these different things. Okay, so here's a basic outline of what we'll be talking about for Chapter 6, and let's get started. Okay, so microbial growth, as you recall from previous labs and lectures, is an increase in the number of cells. It is not referring to the size of the cells. So although it's true that cells do double in size, this is very insignificant. Looking at an individual cell doubling in size right before it divides in half is insignificant, especially when you think of this in light of multicellular organisms like animals and plants, which exponentially increase in their overall size. Moreover, just comparing the amount of microbes over time is really what we care about. It's what's going to actually make a difference in all the different things that we were talking about just a moment ago, all the applications of microbes, the harm that they can cause, and also how we can use them for good, right? So we care about how many microbes there are. A colony is specifically a group of cells, typically on agar, right? And so typically grown on agar plates, or on an agar slant in a test tube. And it's really important that you remember that they all originate from one cell. And so therefore, because it's one cell dividing and then the 2 cells dividing, they're all genetically identical. So in these blanks here, I want you to put genetically identical. Okay. And I underlined one cell so that you remember that one colony originated from one cell, and they are all genetically identical. Okay, there are so many cells in one spot that you can actually see them with your naked eye. Remember, normally to be able to tell if a microorganism is present or not, you would need to look under the microscope. A colony is looking at a mass of so many cells in one spot that you can see it with your naked eye. Okay, so as I already mentioned, although the cells do double in size before they divide in half, that's not the point when we're talking about microbial growth. Okay, so physical requirements. We've already talked about temperature because we started talking about enzymes, remember? And so microbes have a minimum growth temperature that they can survive, an optimum, and a maximum. And recall that the optimum is pretty close to the maximum. And maximum after you reach the maximum, they start to die off because you start to denature those enzymes. So the three main groups of microorganisms as far as temperature goes, so you will see that they're actually divided into 5 groups when we look at this little chart on the next slide. But really, it's 3 main groups that we, or I, care about, and that's the thermophiles, the psychotrophs, and the mesophiles. And so we'll talk about those with, some detail, some things for you to write some notes about. And then on the next slide, you'll see the other 2 groups that I didn't really mention much about because we just don't have a lot of applications for them. So recall that body temperature is 37 degrees Celsius, and of course that's equal to 98.6 degrees Fahrenheit. And Celsius, 1 degree Celsius is much more than 1 degree Fahrenheit. So when you go up to saying 40 degrees Celsius, that might not sound that warm to you compared to 37, but 40 degrees Celsius is actually 104 degrees Fahrenheit. Okay. And so thermophiles actually means heat lover, right? And so we'll see these growing as low as 40 degrees Celsius and as high as 70. So another thing to note about these groups of different types of organisms that live in a range, similar range, is that these ranges tend to be about 30 degrees. So organisms can, on average, tolerate a range of 30 degrees Celsius for the bacteria and archaea, so for prokaryotes, right? All right, so thermophiles, heat lovers, and even the hyperthermophiles, which are even hotter, 95, they range from 70 to 110 degrees Celsius with the optimal temp of 95. So these 2 groups, the hypothermophiles and the thermophiles, are mostly talking about organisms that you find in compost, hot springs, and thermal vents, right? So very hot climates, and they're typically not pathogenic because, well, we're not hot enough. We wouldn't be a good host, would we? If they're the lowest that they can survive is 40 degrees and we are 37. Okay. Now, psychotrophs, on the other hand, like the cold, and their range is from 0 to 30. And though, again, we are not a proper host for them because now we're too hot, they can be significant when it comes to food spoilage. So these are not microbes that are causing disease, but they can make your food go sour, right? So food spoilage. They grow from 0 to 30 Celsius, and a refrigerator is about 5 to 40. A refrigerator is about 5 degrees Celsius or 40 degrees Fahrenheit. And so these psychotrophs can grow in the refrigerator at refrigerator temps and spoil your food. So this is one of the reasons why milk, for example, lasts on average 2 weeks, and then you have to toss it out. All right, mesophiles are the group that we're talking about throughout the course the most because these are where the human pathogens are found, right? There are other non-pathogens in this group as well, but for our purposes, we care about the pathogens. So because the optimal temperature for mesophiles is 37 degrees Celsius, and that is our body temperature, you can see the connection, right? So there's also extreme cold lovers that I didn't mention. And you can see there they can range from -10 Celsius to 20. So each of these has a range of about 30, except for the mesophiles actually range larger, 40, right, from 10 to 50. And then the hyperthermophiles also have a larger range. But, we can say about 30 degrees from min to max on average. I already talked about everything else that I wanted to talk about. So again, I know we don't use Celsius in everyday life, so you need to memorize that body temperature is 37 degrees Celsius, and boiling 100 degrees Celsius. That, to give you a frame of reference, is about 210 Fahrenheit, right? So 100 degrees Celsius is boiling, which is about 210 Fahrenheit. Okay. So temperature makes a huge difference on the allowing organisms to grow. And recall that when things get too cold, you're not killing the bacteria, but you are slowing down or even pausing their growth until it gets warmer. Okay. And if you get too hot, then you actually can start to kill them. So we have, for food safety, we have what we call the danger zone. The danger zone is anything from pretty much room temperature up to about 50 to 53 degrees Celsius, which is pretty warm, right? So remember, our body temperature is 37, so we're smack in the middle of that danger zone. Going all the way up to 130 degrees Fahrenheit is going to feel pretty warm to us, but that's still not hot enough to keep your food safe. So this is why you should always keep your food really, really hot or really, really cold, and never let your food sit out for more than an hour. So you cook it, and then you serve it, and you eat, and then you put it away. If you tend to spend more than 45 minutes to an hour together at the table eating, then you would need to take a break to put your food away and go back to the table, or maybe you put it away immediately once you serve it. Regardless, if you leave it out for more than an hour, you really should dispose of it. Likewise, once you put your food in the fridge, you need to eat it within 3 days. 4 is kind of pushing it, but depending on what it is, how quickly you put it away, you could maybe get away with that 4th day. You should definitely not allow food to stay in your fridge for an entire week, which I know a lot of people are in the habit of doing, and that is taking a risk. Another thing I want to talk to you about is that when you allow bacteria to start growing, depending on which bacteria are present, remember we've talked about like Staphylococcus, for example, and Streptococcus, and how like different bacteria can produce toxins. So if you have just even a few of those bacteria present and they start to grow, they're going to produce toxins. And once those toxins are present, you cannot get rid of them. It doesn't matter how long you cook your food. The next time, you're not going to get rid of the toxins. Even if you kill off the bacteria by, say, putting it in the oven and cooking it for, very high heat, it's not going to get rid of those toxins. Okay, so good hygiene, washing your hands thoroughly, having clean utensils and whatnot during preparation is very important. Okay. Anything colder than this danger zone, you can still, many bacteria are going to survive, and some might even grow, right? Because remember, getting cold doesn't kill bacteria really, it just slows down the growth. Okay, 0 to 5 degrees, which is, around your fridge temperatures, should significantly slow the growth of spoilage bacteria. Anything below 0, you're really not going to see any growth. It's too cold. Once you defrost it, then it can start growing again. And anything above the danger zone, there's about a 10 degree range where you're reaching that, you're surpassing the optimal and passing the maximum, and you're starting to kill off some bacteria. So you get very slow bacterial growth. Some are even starting to die, and the hotter you get, the more you are killing. So another thing when we talk about killing off bacteria is that it's at a constant rate, and it depends on how much organic material is present. So the more stuff that there is, the longer it's going to take to kill off the microbes. Okay, so always keep food super hot or super cold. And another thing I want to talk to you about is that a lot of people, and I've seen this firsthand when I used to rent out rooms, and I had a lot of different roommates. People think that they need to let their food cool down before they put it in the refrigerator. But food's going to cool down much, much quicker once it's in the fridge than it will sitting out on the counter. Okay, if you're worried about having a really large amount of food, then divide it into smaller containers and stick it in the fridge right away. Do not let it sit for more than an hour once it's done cooking. Okay. Reheating in a microwave is not going to kill bacteria. If it kills any, it's not going to kill very many. I'd be surprised if it really killed any at all. The microwave is a short amount of time, and it's typically not even getting very hot. And it would never remove toxins. Okay. The other problem about toxins, right? So you have low level amount of bacteria. They're growing, they're releasing toxins, but there's not a lot of the bacteria present. So you're not going to have any weird smell or taste, nothing that's going to indicate that the food has anything wrong with it. And then, of course, you ingest that, and then you're going to be pretty sick, vomiting, diarrhea, the whole works. Okay. All right. So that was food safety. Now let's talk about pH. Okay, so the reason that we do things like pickling is because most bacteria and even molds and yeast have a range that they can tolerate, right? So bacteria grow best between 6.5 and 7.5. Some bacteria are going to be in a tight range that can only tolerate from 7 to 7.5, whereas others might be able to go from 6.5 to 7, right? So I'm not saying that all bacteria can tolerate the entire range between 6.5 and 7.5, but if you look at the different groups of bacteria, they will fall somewhere between 6.5 and 7.5. And then mold and yeast are a little hardier. They can go more acidic. But it's very rare that you find an organism that can survive something as extreme as something like pH 4. Oh, looks like there's a typo. That should not be capital P, should be lowercase. So stomach acid, being around pH 2, is excellent for killing lots of bacteria because it's extremely acidic and they can't tolerate that. Furthermore, then it goes from being really acidic to being quickly neutralized and even a little alkaline when you get into your gut. And so anything that did survive probably is going to be killed from that drastic change. And then, of course, you do get sick sometimes. You can have gastrointestinal infections, so not everything is always killed. But also need to think about everything you drink and everything you eat is full of bacteria. Your hands are covered in it, the air you're breathing, right? So you're constantly killing lots and lots of bacteria without ever knowing. Okay. Pickling, you can obviously pickle cucumbers, which we call pickles, but there's all sorts of other things that people will also pickle. Is just the act of putting something in a very acidic solution to preserve it. And so it's very effective, right? Because it's under that pH 4. Now, something else I want to remind you of is that there's a lot of cellular waste from metabolism that is producing acidic products, right? Remember last chapter we talked about, for instance, carbohydrate fermentation leading to lactic acid or alcohol or other types of byproducts. And so we see that when bacteria are growing, they'll eventually slow down or even stop increasing in numbers altogether because they start to harm themselves with their waste products. They also are eating up the food, and they'll slow down when food's not as available. Of course, there's exceptions. We always give rules or patterns, and then there's always going to be an exception to those patterns, right? So there's a group of organisms called the acidophiles that actually grow in acidic environments. But again, that's not something that we're concerned about. So vinegar and stomach acid are good for food preservation. Well, stomach acid's good for killing, and vinegar is good for food preservation. And remember, vinegar is acetic acid. And also, when we talked about the cellular waste, this is why when you're growing samples in the lab, you add things like buffers to slow down that acidity of the solution, right? If you didn't add a buffer, bacteria would turn their media acidic fairly quickly. It could be as quickly as just half a day. And then adding the buffer, you can usually get several days out of that media before you need to change it. Why does pH kill? Why does changing the pH, especially in this case becoming too acidic, but also the other direction, becoming too alkaline, would also do it? We don't eat alkaline foods, so that's why it's not really coming up in our discussion right here. But if you get too acidic or too alkaline, you change the ionization of the few amino acids that have those side groups that are acidic or basic. And so you change their behavior. Now you're changing the bonds that they make, which would be ionic bonds. So you're going to break some ionic bonds. You're going to form new ones that weren't there before. And now it's just completely refolded. And remember, proteins function off of their shape. They're very specific on their shape. So you can denature by pH, not just heat. All right. Now, when you denature with heat, you're targeting those weak hydrogen bonds. So it's different mode of action. All right. So we also have osmotic pressure, and the cell wall of the bacteria do help to resist some change in osmotic pressure, but to a limit. Okay, so if you go from 0.85% salt and you go all the way to 10% salt, that's a huge difference, right? So give the cell wall a little credit, but eventually you will overcome its ability to resist osmotic pressure. And what will happen is that the water is going to leave the cell, going towards the salt, or what we call the high concentration of solute. and that causes shrinking, and that will cause the plasma membrane to then detach from the cell wall and, shrivel up. Okay, so the technical term for that is plasmolyzed, not shriveled. I like to make things as simple as possible, and then I have to remember to use the actual correct terminology so you guys can learn it. The terms hypertonic and hypotonic. Hyper, as anyone would associate hyper, like hyperactive, right, as a lot. Like if you're, sometimes we just use the slang hyper. We don't even put the rest of the word with it, right? If someone's hyper, we think, oh, you've had a lot of caffeine, you've had a lot of sugar, you have a lot of energy, a lot, right? That's hyper. Hypo is the opposite. Is slow, or it's less, right? It's the opposite of hyper. Okay, so if you are using these terms hypertonic or hypotonic, you're talking about a solution. You're talking about how much solute. That could be sugar, it could be salt, anything, but how much solute is outside of the cell. Okay, so the terms hypertonic and hypotonic mean in relation to the inside of the cell. You're telling me about the environment outside of the cell. So if you create a hypertonic environment, like making it very salty, you're going to cause the water to go, right, remember this is a membrane, and the salt cannot just cross it cannot cross the membrane, so water has to. Why? Because molecules like to be at equilibrium, and so the water is going to then move towards the higher concentration of solute in an effort to try to make an equilibrium. And so that's going to cause the membrane to pull away from the cell wall as it dehydrates, and this is referred to as plasmolysis. And that's why sometimes salt is used as a food preservative. I know we have other like fancier chemicals that we also use as food preservatives, but the original food preservative is salt, and that still is true even today. Okay. All right. Of course, there's also exceptions to this high salt rule, where we see that there are organisms that prefer or even require high osmotic pressures. So they're adapted to live, in salty solutions. And of course, putting them in something that would be good for this cell would be not enough, and it would actually cause it to burst or lyse open, so it would cause the opposite problem. Okay, so now we can look at some chemical requirements for growth, which we've also talked about here and there. So recall that all of life requires carbon as the backbone of all of the organic molecules. So to be an organic molecule literally means to have carbon as the backbone. And so glucose, DNA, lipids, proteins, everything, all of the organic molecules have carbon as their backbone. This is why we say water and carbon dioxide are inorganic, because they don't have a carbon backbone. Okay. Now, these organic molecules, glucose, DNA, lipids, proteins, they could be structural, just being think of it as like the framework of the cell, right? Just support. Or they could be for actual energy sources, or they could be messengers that get sent throughout the body, right? So there are different functions, but we need carbon to make all of these organic molecules. We have a group of prokaryotes called the autotrophs. Excuse me, it is getting a little late over here. Okay, so autotrophs use inorganic carbon. Instead of consuming organic molecules, breaking them down, rearranging them, they will take carbon dioxide out of the air and use it as their carbon source. So that's what an autotroph means. On average, if you take a bacterial cell, you could, do some analyses and weigh the total amount of carbon versus hydrogen versus the oxygen and all the other elements, and you would see that carbon will make up pretty much half of the dry weight of bacteria. So not counting the water, but half of the dry weight of bacteria is carbon. So it really is the backbone of life. Okay. Nitrogen, is required for all amino acids because every single amino acid has an amine group, NH4 group. Nucleic acids as well, right? So DNA and RNA are made up of nitrogenous bases, right? So a nucleotide, remember, is a phosphate group attached to a sugar, and on the other side of that sugar is that nitrogenous base, either A, T, C, G, or U. And so in order to have a nitrogenous base, you must have nitrogen. And most bacteria are actually able to break down or decompose proteins and use protein as a nutrient source. So catabolic reaction to decompose proteins, right? Some bacteria can even use ammonium, NH4+, or even nitrate, NO3- as their nitrogen source. Very few bacteria can use nitrogen out of the air, molecular, right, into, and that's what we talked about since day one, these very special bacteria called the nitrogen fixers. Because if we didn't have bacteria that could do this, then we wouldn't have trees and we wouldn't have any life on Earth, right? Okay. More chemical requirements. Sulfur, also required for some amino acids, not every single one. We also have some coenzymes like biotin, and this is a repeat, but most bacteria can decompose proteins and they would be able to get sulfur from there, but some bacteria can use inorganic sulfur sources. Phosphorus we find in pretty much all of our molecules that we've been studying, right? So we see DNA and RNA have that phosphate group, ATP, right? Triphosphate, membranes of all of our cells, phospholipids, right? So phosphorus is very numerous, right? We need it in large amounts. And then we also need things like potassium, magnesium, and calcium. We need them for cofactors for enzymes. Multicellular organisms also use these as electrolytes to conduct electrical currents throughout the body and to balance our water in our body. And that's why when you're dehydrated, you need to replace your electrolytes and not just water, because the electrolytes play such an important role in the body. Okay. Trace elements means we need them in very small amounts compared to all the other things that we've been talking about so far. These are inorganic elements like iron, zinc, copper, and these are also usually used as cofactors for enzymes. You also know that you need iron for your hemoglobin, right? Okay, toxic oxygen species. This is, perhaps one of the slightly difficult topics. Try to make it a little easier for you, hopefully. So the oxygen in the air, O2, from pretty much from the sunlight, can become kind of excited and go into this higher energy state, and it can form what we call free radicals. And so, one of the most reactive types of free radicals is called the hydroxyl radical, OH, with this little dot indicating that it's a radical. And so it's a natural occurring free radical, due to the ionizing radiation of the sun. Whereas we'll see like also the singlet oxygen. So this is another type of free radical that also comes from just normal molecular oxygen. And then we also have this other type of free radical called the superoxide radical, which is O2 minus. And really this 2 should be small, right below, and then the minus should be superscript up above. It looks like maybe some of my formatting didn't save, unfortunately, so you'll just have to correct that on your notes. Okay, so superoxide radicals, which is another type of free radical. This is a natural byproduct of aerobic cellular respiration. And you saw this at the end of the electron transport chain, right? So we have to have enzymes because nothing's perfect. We show these diagrams in the book, and it looks like it's all, it's always this way and it's perfect. But of course, nothing's perfect. So you can have some of these free radicals not get dealt with immediately, and because these all of the free radicals are highly reactive, as soon as they come into contact with a molecule, it's going to donate that extra electron, and then that's going to just disturb that molecule and make it reactive, and then it's going to do the same thing to the next molecule it contacts, and so it acts as like a chain reaction. And when this happens within a molecule, it actually can cause bonds to break and reform, and they don't always reform as they were originally. And so you can get mutations in your DNA, for example, this way. Now we have, as humans and most organisms, have a DNA repair system, and so we can cope with certain levels of damage. And then if you have more damage than you're able to cope with, then that's when you actually are putting yourself at risk of things like cancer, for example. So anyways, bacteria, if they are undergoing aerobic cellular respiration, just like any organism, has to have an enzyme to deal with these free radicals that they're making as a side effect of respiration, because free radicals are very damaging. And you've heard people talk about free radicals and needing to have antioxidants to deal with them. And antioxidants indeed are molecules like vitamin C, vitamin E, that can they can actually take on that extra electron and stop the process, stop that chain reaction. It ends it. But we also have enzymes called superoxide dismutase, abbreviated SOD, which are very effective for dealing with these free radicals during cellular respiration. And so they neutralize the radicals into hydrogen peroxide, which sounds great, like, oh, okay, problem solved. But not really, because once you have hydrogen peroxide, you have to realize, well, hydrogen peroxide is also reactive, a little toxic, not as extreme as the free radicals, but still enough to where we don't want it lying around, because it will dissociate in solution and form the peroxide anion. And that's going to be reactive enough to cause damage as well. So we need another enzyme, such as catalase or peroxidase, to deal with the hydrogen peroxide that's being made from the superoxide dismutase. Okay, so both of these are going to break the hydrogen peroxide down into water. Catalase produces water and oxygen gas, and that's how you get the bubbles. So when you have a cut and you pour hydrogen peroxide in your cut to get rid of any debris and to kill off some bacteria, you're going to kill bacteria that don't necessarily have the enzymes to deal with it. But the reason that you get those bubbles that form in your wound that you pour the hydrogen peroxide on is because you have lots of catalase enzyme within your blood and your cells. And so you rapidly turn the hydrogen peroxide into water and oxygen, because it would also be harmful to you, to your own cells, if you didn't have catalase. Okay, so that's kind of cool, kind of fun example. You can, I'm sure everyone is familiar with, but if not, you can look it up on and watch it on a YouTube video if you want. And so some bacteria have superoxide dismutase and catalase. Others have superoxide dismutase and peroxidase. And furthermore, some will have all three, and they'll be really good at dealing with toxic oxygen species. And it's really important that you start thinking about bacteria in relation to how they cope with oxygen, whether they can or not, and how well they do. And so that's where this diagram comes in. We have several groups of bacteria based on how they grow in relation to oxygen. So the first thing you need to note is that these tubes are showing you a semi-solid media, and it's a special type of media that all the oxygen has been removed. And so you can see, you'll know, okay, so you've removed all the oxygen from this media, and from the properties of oxygen, we know oxygen does not diffuse very well through the media, because oxygen is poorly soluble in water. So because oxygen's not good at dissolving in water, it's only going to diffuse at the top of the tube. So you're going to have the most oxygen at the very surface and less, and then by the time you get to the middle, it's pretty safe to say that you don't have any oxygen, very, very little, and then none at the bottom. Okay, so no oxygen at the bottom. The most oxygen that you will have will be at the very top, okay? And then you get like a nice little breakdown or concentration gradient. That's what I was looking for. Okay, so we have a group, our first group of microbes we're going to talk about in tube A, called the obligate aerobes. They must have oxygen in order to survive, grow. So these are performing aerobic cellular respiration, where oxygen is the final electron acceptor, and they don't have any backup. That's all they can do. Okay, they, however, are really good at dealing with toxic oxygen. They have the SOD and the catalase enzymes to deal with the oxygen. Okay, tube B, on the other hand, is the exact opposite. It's an obligate anaerobe. It only grows in the absence of oxygen, all the way at the bottom. And so these organisms don't have SOD, they don't have catalase, they don't have any enzymes to deal with oxygen. That's why they will be killed if they are exposed to oxygen. Okay, they grow at the bottom of the tube where there is no oxygen. And they could be undergoing anaerobic respiration, right? Where they have something other than oxygen as the final electron acceptor. Okay, then our next group, facultative anaerobes. Facultative means that they have the ability to do both. Okay, so facultative anaerobes can use anaerobic means as a backup. They prefer oxygen. They grow the best when oxygen's around. So aerobic respiration is their preference. And you can tell because it's much more dense at the top where the oxygen is. And you have fewer and fewer, but you do get some growing throughout the rest of the tube. And that's how you know it's facultative anaerobe. So because it's very common for oxygen to be limited, as I mentioned earlier, oxygen does not dissolve well in water, and so throughout the earth we have lots of environments that have very low or even no oxygen at all. And so there are a lot of microbes that have adapted to this, and they are able to switch on either anaerobic respiration or even fermentation. Okay, so many bacteria and even yeast are facultative anaerobes. Facultative anaerobes grow best with oxygen, but can survive without oxygen. Right, they can indeed survive without oxygen. Yeast and some bacteria will only be able to go to fermentation, which you know is a lesser, you get less energy that way. You'll go through your food source faster and get less energy from it. Most bacteria, however, will use anaerobic respiration. For example, E. coli. Okay, and they produce enzymes, right? The SOD, the catalase. Okay, and then we have D, and D is an aerotolerant anaerobe, meaning that it produces SOD enzyme and only SOD enzyme. It does not use oxygen at all. So it's using anaerobic respiration, for example. Never going to utilize the oxygen. But because it produces the SOD enzyme, it's also not harmed. So it can grow anywhere. It doesn't need oxygen, it doesn't use oxygen, but it doesn't care if oxygen is present. So this is your, like, if you want to give, personalities to bacteria, which of course is, fictional, right? They don't have personalities, they're single cells, but it's fun and easier to remember things when we do. And so I would think of these as the easygoing, right? They don't care. Put me in a lot of oxygen, take away all the oxygen or anything in between, and I'm fine. Go with the flow. Okay, that's the aerotolerant anaerobes. On the other hand, the Goldilocks, the picky bacteria, are the microaerophiles. They need oxygen, but they cannot handle it if there's too much, right? So you have to give them just a little bit of oxygen. And this is because they do use oxygen for, aerobic respiration. Oops, sorry, I accidentally hit my keyboard. But they're sensitive to the superoxide radicals and the peroxides. And we don't really know for sure. We know that they're sensitive to these free radicals, that the very radicals they're producing by their own respiration and by being exposed to oxygen in the air. But they must make the enzymes, or else they wouldn't be aerobic at all, right? So my assumption is that they probably just produce lower levels of the enzymes than the other aerobic bacteria, and so that's why they're sensitive. So basically, like, they just don't make enough of the SOD and catalase enzymes to to tolerate the normal amount of oxygen. And for that reason, they're also going to grow a little slower, right, than some of the other bacteria that we see. All right, so this has been your overview of oxygen requirements. So we've still been talking about the chemical requirements for microbial growth, and all the other slides looked kind of, like this, where we talked about, carbon, right, nitrogen, sulfur, phosphorus, etc. And then we get to oxygen and it looks a little different. So I just want to remind you that this is another one of the chemical requirements for growth. And for some of them, the requirement is that there's no oxygen, right? It's obligate anaerobe. And for others, it requires oxygen. And then we have the other situations. So you have to remember all of these different types of bacteria and what their relationship to oxygen is. Okay, organic growth factors would be things like vitamins, amino acids, purines, and pyrimidines. So what are purines and what are pyrimidines? They're the sugars, or the rings rather, for your nitrogenous bases, so A, T, C, G. Okay, and purines are the big ring, and pyrimidines are the smaller ring, if you were to see the images of the nitrogenous bases. But that's not important. I'm not going to test you on the chemistry of those. But purines and pyrimidines are the different types of nitrogenous bases, rings, though. And then, so an organic compound is just one of these molecules that is carbon-based, carbon backbone, and we call it an organic growth factor when it's essential. I don't know why they don't just call it essential, but we want to specify that it's organic. Okay, so the organism is unable to synthesize some particular nutrient, and it'd be called an organic growth factor in that case. Each type of organism has its own requirement, meaning it can synthesize different things. So I cannot give you a list of what are the organic growth factors. It's going to be different for each organism. For humans, for example, we have 13 essential vitamins and 9 essential amino acids. So there you go. That's a lot of things that we have to consume in our diets. And so you can imagine then bacteria are going to have some essential organic nutrients as well. Okay, so I think that's a great time to stop. Please make sure that at the beginning and end of these sections, you're reading these learning objectives and check your understanding questions. Okay, they're a very helpful tool for you to study with. All right, so you're going to read about culture media on your own. You've already been introduced to these ideas previously in lab in particular. So make sure you're able to distinguish between what we call chemically defined media and complex media. And what does it mean for an organism to be fastidious? So those are going to be new terms that we haven't talked about yet. But something we did already talk about is how do you obtain a pure culture? So you can refresh yourself on that concept. And then another thing I want you to read that is new are the different types of anaerobic growth techniques that are presented in your textbook. Okay. And then review selective and differential media, right? We talked a lot about that already. And then another new term is enrichment medium, or media is plural. Okay, so what does it mean and when would you use enrichment media? Okay, so some review and some new content for you, but you're going to read your textbook to do that. And then make sure that you can answer or do all these things. All right, so that's the end for this video, and then I will post another video probably tomorrow. All right, good job.